Hello and welcome dear viewers to the video on photostability study and stabilization of pharmaceuticals. In this video we are going to see what is photostability study, how it is performed, how the results are interpreted, and what are the measures taken for the photosensitive or photolabile drugs and the formulations. How these photosensitive drugs are manufactured and how the formulations are stabilized. So as the light has degradant impact onto some of the drug substances and formulations, the degradation will happen and there will be increase in the impurity level. As the related substances or organic impurity is one of the critical quality attribute for the pharmaceutical formulation, and also it is one of the most important critical material attribute for drug substances. The photostability study becomes very much important. As the formal stability studies are performed for the formulations, these photostability studies are also performed. Photostability study is performed on drug substance and also onto the drug product, that is the formulation. The study of effect of light onto the quality parameters of the API, that is drug substance, and the drug product, that is known as the photostability study. And this study is to predict the sensitivity of the drug substances and the drug products for the effects of light. These studies are performed as a part of force degradation study also, and also as a confirmatory study for the pharmaceutical formulations. And if there is any sensitivity found by the photostability study of the API or the product, then the product is called as photosensitive or photolabile, and then the necessary actions are required to be taken for its protection from the effect of light. The light exposure or the light may increase the degradation, and that's why the exposed samples of the API or formulation may have higher impurity limit. So mainly the degradation is required to be studied. The photostability studies involves the effect of light with appropriate exposure on the physical and chemical properties, and thus effect of these changes onto the safety and efficacy. So not only the chemical changes but also the physical change, chemical change is studied. And the main guideline which deals with the photostability study is ICH Q1B. It is the quality guideline series and it is one of the guideline from ICH guideline, that is Q1B. This guideline gives the clear information about the photostability studies. So now the objectives of photostability studies are two. First is force degradation study and second is the confirmatory study. These studies are performed to check whether the drug substance or drug formulation is photosensitive or not. The force degradation by the light effect to elucidate the impurity formation by light effect or for method development and the validation. So in the early development, force degradation study is performed, and in that force degradation study, the effect of light is studied. This study is required for understanding the stability indicating nature of the method. Also, it helps to understand the impurity formation pathway due to the effect of light or exposure of light. Some studies for the force degradation study also involves the effect of direct sunlight direct UV light and also the effect of indirect UV light and indirect sunlight. So based on that, the studies are performed. One thing here to understand is that though there is a degradation due in the force degradation study due to light, we cannot say that the product or the drug substance is photosensitive. For that, we should perform the photostability studies as per the ICH Q1B guideline. And in that study, the specific exposure for the light is given. And if the product or drug substance found to be getting degraded or if any CQA is getting changed, then we can say that the formulation is photosensitive. But from the results of force degradation study, we cannot say the formulation or API is photosensitive. The reason behind this is that the force degradation studies are harsher compared to the actual photostability studies. Then is the confirmatory testing. The testing of product or for light sensitivity and appropriateness of packaging system to protect from light. So confirmatory testing is done. And this testing will give you clear information about the photosensitivity of the product and also whether your packaging system is capable for protecting it from the light or not. Recommendations for manufacturing areas like use of sodium vapor lamp, in process storage area, testing area lighting, use of sodium vapor lamp, use of testing glasswares with protection to light or amber color can be used if you found the drug substance is photolabile or the formulation is photolabile. Then design of the packaging system and labeling and storage recommendations like protect from light or use of amber colored bottles can be decided based on the confirmatory photostability testing. Now, how the samples are prepared for photostability testing. So for drug substance, API is put into the Petri plate and it is sprayed. Generally, the layer thickness is around 3 millimeters, 3 mm, and along with it, the control sample is kept. That is known as dark sample. In simple way, the dark sample can be put or should be put with the exposed sample so that you can compare the effect of light onto the dark sample and the exposed sample. The dark sample can be prepared by wrapping it in aluminum foil or covering it fully, or you can store the API sample in alu pouch so that light exposure will not be there and you can compare the effect of light. Then drug product. So if you are testing tablet and capsules, so put some tablets and capsule in petri plate and don't cover it. It should be open. Product in primary pack, that is blister pack or strip, or you can put the bottle. Product in the secondary pack, like blister in carton or the intended market pack is required to be kept. Similarly here, control sample or dark sample can be put by wrapping in aluminum foil or fully covering it, or aluminum pouch can be used. Also, some of the applicants keep API with drug product so that the effect of light on API and effect of light onto the formulation can be compared. So that can also be done. But generally, when you are generating the data for filing, that time these practices should be avoided. Then placebo is also kept for comparison. So this is regarding the drug substance and drug product. In research phase, all these samples are required to be tested and along with the drug product, the drug substance should be loaded so that all the samples will have the same exposure and at a single run these samples can be analyzed. The light exposure requirement is given as per the SHQ1B guideline and gives the exposure of not less than 1.2 million lux hours. Lux is a unit of light and the exposure should be not less than 1.2 million lux hours and an integrated near ultraviolet energy of not less than 200 watt hours per square meter. So it is the not less than 1.2 million lux hours, white fluorescence, white light and not less than 200 watt hours per square meter of near UV light. The light sources may be single or may be separate. The lamp to provide the visible and UV radiation or light. The example is D65 lamp, ID65 lamp and UV range of 120 to 400 nanometer is required. Separate lamps can be used generally for the illumination and the exposure time calculation is required so that you can calculate the hours exposure. For example, the lux per hour calculation is required. If the light source gives the 12,000 lux per hours, then you will require 100 hours for getting the illumination of 1.2 million lux hours. Similarly, watt calculation can be done. 2 watt lamp can be run for 200 hours to get 200 watt per square meter. Here 2 watts lamp can be run for 100 hours so that you can get the 200 watt hours per square meter as a light exposure. Then confirmatory studies sample should be exposed to light providing an overall illumination of not less than 1.2 million lux hours and an integrated near UV energy of not less than 200 watt hours square meter to allow direct comparisons to be made between the substance and the product. The sample should be tested before loading into the photostability chamber, that is called as initial testing, and it is also required to be tested after completion of the exposure. The API is required to be tested for all the CQAs like description, assay and impurities. Other tests can be included as required and drug product is required to be tested for description, assay, DT time, dissolution time if tablet and capsule product is there, impurities that is degradant and any specific impurity as required. Then the results. Once you prepare the samples, you tested it at initial level and then the photostability charging is done and after completion of the illumination these samples are tested. So results will be acceptable if the product meets the defined specifications, there is no degradation, and the results will be acceptable if the product or the API is not found photosensitive or photolabile. And if it is not acceptable, that is degradation observed, then what is to be done? That time you can say the product is photolabile or photosensitive. And this is regarding the open samples. If the sample is failing in the open condition or exposed condition, then check and compare the packed samples. Like you can check the immediate pack and the marketed pack. For example, for tablet formulation, the blister pack will be immediate pack and the marketed and the blister pack in the carton will be marketed pack. So compare all these and then if you found that if the packed sample is not failing, then you can conclude that the product is passing and the packaging system is protecting the formulation from the effect of light. The second condition may be there if the packed sample is failing. That time the packaging system or formulation is required to be redeveloped. That time you should change the packaging system, like use amber colored blister or bottles, or the formulation changes can be done. The label should indicate that the product should be protected from light or stored in the dark place and in the original container. If the product is passing with the immediate pack or marketed pack, and if it is failing in open condition, then labeling instruction should be given, like store in the original pack. Sometimes if the core tablets are there and the core tablets are packaged into the clear blister, both the samples are failing. Consider a case that both the samples are failing. That time you can go for the opaque blisters, so that you can protect the formulation from effect of the light. The examples of photosensitive or light sensitive APIs are like ciprofloxacin, simvastatin, nifedipine, glipizide. Many of the APIs are photosensitive, and that's why the amber color packaging is used in the pharmaceutical formulations. The clear pack generally allows the light to pass through it, and that's why the light effect has the effect onto the formulation. The amber color pack inhibits the passage of light of wavelength of less than 470 nanometer, and thus it can protect the photosensitive APIs, and thus it has a stabilizing effect. One thing here is to understand that how the degradation occurs due to the light. So as the temperature has effect onto the degradation, similarly light also can induce degradation, and that degradation may be due to the photo oxidation or it may be due to the photo degradation. Then the exposure time will have the impact onto the degradation. Then the source of light, the wavelength of light, all these have the effect onto the degradation. Then if you found that the API or formulation is photosensitive, then what are the ways to protect the photolabile drugs? So these drugs are required to be protected from the initial level. Protect the light, protect the formulation and the APIs from light during the manufacturing, storage and distribution. Sodium vapor lamps can be used during manufacturing and packaging operation, and these sodium vapor lamps generate the wavelength between 500 nanometer to 800 nanometer, and that's why these don't have the degradant impact. Then pack the material in opaque packaging. Opaque means this doesn't allow the passage of light. Storage instruction should be there as protect it from light. Use opaque blisters and bottles for the formulation. Film coating of tablet can be done with the TiO2 as a opacifier in the coating composition. Titanium dioxide is a opacifier and it doesn't allow the passage of light. So if the core tablets are found to be photosensitive, then go for film coating. Also, do not use clear capsule shells. Use the capsule shells with TiO2 as a opacifier, and that opaque empty capsule shell can be used to fill the photosensitive APIs and the formulation. Use amber colored bottles for packaging of the formulations which are photosensitive. Use amber colored glasswares during testing and analysis. Sometimes it is observed that during testing the drug may get degraded, and that's why the results will be there with a higher degradation. So to avoid that, to have the actual results of the degradation or to have the actual analytical results, use the amber colored glasswares, and also if possible to avoid the effect of light, wrap the testing solutions where the drug is used in the aluminum foil. So these are some of the packaging materials. These are opaque blisters. These are also opaque blisters. This is opaque HDPE bottle. This is alu blister which is opaque and doesn't allow the light passage. This is amber colored ampoule for injectables. This is amber colored blister for tablets. So these materials can be utilized, and by the proper instruction during the manufacturing and packaging, we can stabilize the formulations which are photosensitive. So this is regarding the photostability studies and stabilizing the photosensitive molecules. In the industry, in the manufacturing area, the sodium vapor lamps are installed by default nowadays, so that whenever there is a product to be manufactured which is photosensitive, that product can be manufactured in that area. Also, wherever the transparent windows are there, glass windows are there, these windows are required to be covered with black material or black sheets, so that there will not be any exposure of the light, and you can protect the photosensitive APIs during manufacturing itself. Also, the labeling should give the clear instruction to protect the formulation from light. When you are dealing with the photosensitive APIs and photosensitive formulations, the professionals involved in the handling should be trained, so that they will understand the effect of light onto the formulation, and accordingly they can take care for the formulation and the processing. I hope you might have got good learning from this video, and I will request to subscribe this channel to have the notifications for new videos. Kindly do like, share and subscribe to this channel, and thank you for your time.